infection. She has active research laboratory with undergraduate students. And Brianne earned her BS in biology from Duke University and a PhD in immunology from Harvard. I would like to thank the ABRF for their support of this webinar, as well as the American Society for Virology Chat with a Virologist program. So I'll go ahead and turn it over to Brianne.
All right. Thank you so much, David. Thanks for inviting me. Just because you started the TWIV way of doing things, it is currently 52 degrees and sunny in Madison, New Jersey. As David mentioned, I'm an immunologist, and I'm interested in the immune responses to viral infection. I also am part of this podcast, This Week in Virology, where we've been doing episodes about SARS-CoV2 since January. And that has sort of given me a nice perspective on big picture of what's been going on research-wise. And so today I'm going to share with you, given that you are an antibody interest group, some information about SARS-CoV2 antibody responses, both what we know and some of the questions about what we don't know. Just to start out, I'm going to tell you a bit about some general background, just to make sure that we're all on the same page with things that I'll be hitting later. Then we'll talk a little bit about antibody responses to natural infection with this virus, and then some of the big open questions and what we know about those questions. And then we can close up with some discussion of the use of antibodies. And also, as David mentioned, I'm at Drew University, which is an undergraduate institution. So all of my work in my research lab is with undergraduates. I'm very used to fielding all sorts of questions while I'm giving talks, and I would be happy to answer any questions that you all might have as I'm talking. So this whole COVID-19 thing first started in an area of China in late 2019. So it first came onto a lot of people's radar on New Year's Eve, when a report of 27 patients with pneumonia of unknown cause was reported in Wuhan, China. There are records of cases earlier. This says December. There are some discussions of cases in October and November as well. Those patients, as I said, had pneumonia, and it was pneumonia of no known cause. There was metagenomic sequencing, and no pathogens that were known were found in those patients. And in fact, instead, a novel coronavirus was discovered. And so when this naming finally happened, this was named as COVID-19, or the coronavirus infectious disease, that started in 2019. One of the things that I think is really important in terms of the timeline of all of this is that the virus sequence was published in January. So these first patients were recognized less than a year ago. Within about a month, the virus was sequenced. And so everything that's been happening in this outbreak is just incredibly fast and is breaking records all over the place. This virus has some symptoms that we all know about, like high temperature, cough, and breathing difficulties. But we do see all sorts of different symptoms in COVID-19 patients. It's a very heterogeneous disease. And so there are patients with fatigue, there are patients with joint problems, there are patients with loss of taste and smell. There are patients with GI issues. There's a really wide array. What you will also notice is that there are no symptoms that are seen in 100% of the patients. You can see fatigue here in about 80%, but there is no one defining symptom that we see in all patients. And when we look at the symptoms, we have a little bit of a spread of what's going on. So generally, when patients are infected, they may start to show symptoms within about 5 days. The range is about 1 to 14 days. 30 to 40% of those patients won't show any symptoms at all. In total, when you add up the asymptomatic and the mild disease patients, that's about 80%. In addition, you'll see about another group who will have severe disease within the next couple of weeks. That is usually defined as requiring hospitalization and particularly requiring ventilation in the ICU. And then there is another cohort of patients, about 5%, that will go into respiratory failure, septic shock, multiple organ dysfunction at an even later time point, and that may pass away. And so what you should notice here is that there is quite a bit of heterogeneity, again, in the outcome that we see in a number of different patients. And I'm going to mention this again later. That's why I'm sort of making it clear now. We also think that there are some interesting phases of this disease. So typically within the first week or so, patients are in what's known as the viral phase, where we tend to see a fair amount of viral replication and damage due to that virus. Following that, we'll see patients with symptoms that seem to be due to cytokine storm as part of a cytokine storm phase. Following that, and we're learning more and more about the fact that there is a coagulation phase where patients are showing hypercoagulation and really problematic damage to vasculature in the heart, and then potentially a late hyperinflammatory phase. And the reason why I mention this is that in some of these later phases, excess immune responses may be playing important roles in the pathology of disease. And when we think about therapeutics for this virus, we need to think about which phase a patient may be in and which therapies might work best. A therapy that is combating viral replication, for example, might work well during the viral phase, but perhaps is not going to help our patient and may even harm our patient in a coagulation phase. And so we need to be thinking quite a bit about these phases as we're thinking about how we are using different therapies. So all of this drama, all of our staying home and all of the problems that we're currently seeing are the result of this virus, SARS-CoV2. And so it is shown here. It's, of course, part of the coronavirus family. The coronaviruses were so named because under an electron microscope, they look like they have a sort of like the sun's corona, or like they have a crown because of their spike. This virus has a genome size of 29,881 base pairs. So this is actually basically around the size of some of the largest RNA viruses. Coronaviruses are the largest RNA viruses. And have some unique genome replication processes because their genome is so big. There are a number of different proteins that are really important that are part of this virus. And so you can see that there are proteins 1A, ORF1A, ORF1B. You can see there are things like ORF3, M, 6, 7A, 7B, as well as a protein listed here as S, or spike. This spike is shown on the virion below. And the spike is the major determinant of cell entry, as well as a major target of antibody responses, as I'll show you in a bit. Another protein that is particularly important is the nucleocapsid protein, which complexes with the RNA and basically coats the RNA. And this protein is also pretty important immunologically, simply because of the copy number of this protein that we see. Spike gets a lot of attention when we think about immune responses to this virus. And I just wanted to point out, because this actually knowledge really changed my thinking in a couple of ways, that the spike protein itself is 1,273 amino acids long. So the spike protein is a huge protein. and is perhaps bigger than a protein that you might think about on a normal day-to-day basis. That spike is responsible for binding to the receptor to allow the virus to enter cells. Specifically, the spike has a portion that is shown here in the middle, known as the receptor binding domain, or the RBD. The RBD is what actually makes contact with the cell's protein ACE2 in order to allow the virus into the cell. And the RBD actually has to undergo a conformational change to go from a down conformation to an up conformation in order to make that, that interaction happen. And so those are also things that end up being rather important when we think about some of these responses to this virus. The one other really important thing that comes up again and again when thinking about this virus in terms of kind of background information is the phylogeny of coronaviruses. And so coronaviruses are, as I said, are a whole group of viruses. There's not just this one coronavirus. They are in a number of different lineages. Here you can see the alpha and beta lineages, although there are also other lineages. And there are a few other viruses of interest that are within some of these groups. So there are, in fact, 6 other human coronaviruses that infect humans at some point or other. 2 of these are in the alpha coronavirus group. They have really descriptive names, 229E and NL63. They both are causes of common colds in humans. There are also 2 other human coronaviruses, HKU1 and OC43, that are in the beta coronavirus lineage that also are human common cold coronaviruses. In addition, we have MERS coronavirus and SARS-CoV1, SARS Classic, which have much higher death rates and cause much more severe disease. There are lots of coronaviruses that are endemic in bats. You can see bats depicted throughout this phylogenetic tree. You can also see that there are some pretty important coronaviruses agriculturally, some that infect pigs, like porcine epidemic diarrhea virus, PEDV, that's shown here. There's a mouse virus that is pretty frequently studied in research. There's also a cat virus that is not on here, called feline infectious peritonitis virus. And so there's a pretty wide range of viruses within this family. The reason I mention this is that some of what we know about immune responses when we're thinking about SARS-CoV-2 is gleaned from comparisons to immune responses with some of these other viruses. SARS-CoV-2 is also in the beta coronavirus lineage, lineage B. It is almost entirely genetically similar to the bat virus that is listed on this slide. And you can see that it is quite closely related to SARS-CoV-1, as one might have expected. You also may have heard of a newly emerged virus of pigs, SADS coronavirus. It's in the alpha coronavirus lineage. And we think that there are many, many, many viruses within this group that are yet to be described. We see members of this group infecting amphibians, mammals, birds, pretty wide array of organisms. So all of that is fascinating to me as a virologist and a viral immunologist. But of course, you all are a group who is an antibody interest group. And so, of course, your interest is likely going to be about what kind of antibody responses we have to that infection. Happily, there has been quite a bit of study of exactly that question and trying to figure out exactly what's going on there. And investigators have been able to determine epitopes that are recognized by patient antibodies. This is some data from an assay called VirScan that uses both 56 MERS and 21 MERS to test patient sera and look for antibodies recognizing different proteins or different epitopes from individuals. And in this study, they used all of the different SARS-CoV2 proteins and looked for antibody recognition using both COVID-19 patients and controls whose serum had been taken before COVID-19. So we know that they would not be positive and would not have this virus. And you can see that the majority of the antibody responses seem to be against the spike protein. This very large protein that is the transmembrane protein is also important in the attachment. Sort of makes sense that this would be a major antibody target, as well as the nucleoprotein, which is particularly abundant protein in the virion. There are small numbers of positives to other proteins. So actually, if you're looking at this, you can imagine a whole bunch of really skinny columns, each column being a patient. So in this left section, there are 232 columns, each column being a patient, and we're looking to see whether or not there are hits in each protein. So you can see that a patient here or there makes antibodies that will recognize the membrane protein. A patient here or there makes an antibody that will recognize ORF9b. But the vast majority are to nucleoprotein and to spike. You can also notice that there are some antibody responses to the ORF1 protein, but those antibody responses are in fact quite similar to antibody responses to that protein that are seen in the uninfected controls, which led these authors to conclude that these may be antibodies that are cross-reacting with some of the other human coronaviruses. And so the authors of this work sort of discounted those ORF1 antibodies as being as important in this study. And others have been able to use other techniques, in this case, ELISA, to try to measure those antibody responses in infected individuals. And they were able to see IgG and IgM responses to the receptor binding domain, RBD. They looked both in some controls that are shown here in black and a number of different patients, which are shown in gray, at the antibody dilution over time. It's basically antibody titer, as well as the area under the curve of those curves. And so you can see that the patients had IgG and IgM in response to the receptor binding domain, as well as in response to spike, kind of as would have been expected. And in this other study, they also did ELISAs looking for antibodies against RBD, and they saw IgG, IgM, and IgA. So as I'll mention again a little bit later, the observation of antibodies against these antigens with multiple isotypes may be particularly interesting and of particular importance. One other piece of information that I just want to point out quickly here is that in these studies, just sort of assessing the immune responses of some of these infected individuals, there were a few other strange things noted. And one of them is shown here in this part G, where the percent of CD19 positive cells, the percent of B cells in these patients were examined. And you can see that the COVID patients seem to have fewer B cells, which may sort of bode poorly for their antibody response. And again, I'm going to come back to mention something about that as I go forward. So you can see that even from some of the beginning sort of descriptive types of work, there were hints that something strange might be going on. These assays really were looking at antibody presence by ELISA. Other groups have looked at neutralizing antibodies in infected individuals and have been able to detect neutralizing antibodies. Here, they were looking at how well virus replicated in the presence of antibody. And so with the controls, the virus replicated really well. Whereas as the antibodies were added, virus replication was blocked. And so neutralizing antibodies could be seen in a number of different patients, which was exciting. These data were a little bit tricky in the field. I don't know if I'll go as far as saying they were controversial, but they were certainly discussed a bit because many of these assays first used pseudotyped virus instead of using actual SARS-CoV to look at neutralization. That, of course, is due to the requirement for BSL-3 facilities for using SARS-CoV2. And so this is an example of one of the pseudotype neutralization assays, which some people were arguing a little bit about in the past. But in any case, neutralization has also been seen. One of the things that has been really interesting to me when I looked at some of these antibody responses is shown here. In this study, the investigators took B cells that were making RBD specific antibodies from a number of different SARS-CoV-2 infected individuals. So they have both their control individual as well as 6 different patients that are shown here. Obviously, given that they go up to number 107, they had a whole lot more than 6 patients, but we're just showing 6 here. And those B cells were isolated, and then the antibody combining region was sequenced. And so both the Ig heavy and the Ig light region were sequenced. And so you can see the antibody encoding region from patient 21, from 57, from 107, another one from 21, from 72 shown here. And one thing that was really interesting was that the antibody responses in the infected individuals were highly convergent. So that you can see here 3 individuals used exactly the same V, D, and J segments in their heavy chain, as well as having rather similar CDR3s and even similar length CDR3s in the heavy chain, and similarly use the same V and J segment in the light chain, and similar CDR3s with even the same very similar length. And so it seems as though there are some very specific ways that the immune system is going to be able to make antibody responses to this virus. And we may see particular epitopes and particular antibody types being used again and again in many different individuals. There were a flood of papers coming out that looked at different aspects of these antibody responses. And one place where those papers varied was in their patient cohort. Some of those groups looked at controls versus COVID-19 patients, while others defined their patients somewhat differently. And so in some cases, they were talking about symptomatic. Sometimes they only looked at hospitalized patients. Sometimes they were looking at patients who'd recovered and were in the convalescent phase. They varied a lot based on timing. And so there were a few places where some of these studies seemed to disagree a little bit. And one thing that was observed that also brought up some questions is shown here. In these data, patients who were known to be COVID-19 positive were examined, and this was based on PCR. And so there were individuals who were symptomatic as well as those who were asymptomatic, but they were all known positives. And their antibody levels here, I'm showing ELISA. There's also neutralization data in this paper. Is presented right here. What you can see is that the symptomatic patients seemed to have higher antibody levels, both in the acute phase, which is shown on the left for both IgG and IgM, as well as in the convalescent phase later on. And this is quantitated in a different way in terms of proportion of patients on the right. And so it's notable here that the symptomatic patients had higher antibody responses. And this is one of those things that my graduate school PI and I would have had arguments about because we would have decided that this was both surprising and unsurprising. You might imagine that symptomatic patients perhaps have a higher viral load. If they have a higher viral load, you might imagine that they would have more antibodies induced. However, we actually have seen in other studies that there isn't a big difference in viral load between these groups. And so it's sort of an interesting question of why is it that those asymptomatic individuals seem to have lower antibody levels? You might think that antibodies would be particularly protective in these individuals. And you might expect that perhaps if you have a lot of antibodies, you will fight the virus very well and you will be asymptomatic. And so this is a little bit curious in that we are noting that the symptomatic patients seem to have higher antibody levels, though of course there are perhaps a million different ways you could imagine that happening. And so this in and of itself, it just tells us that we should keep our eyes open. Similar data have been seen in other studies as well. So this is that same VirScan study with a large number of different individuals, either 190 pre-COVID controls, and so we've got 190 columns here, as well as 131 non-hospitalized COVID patients and 101 hospitalized patients. And we can look at how many peptides they are recognizing in terms of antibodies in their blood samples. And what you can see is that the severe patients seem to be recognizing more peptides with their antibody responses. And so it does seem in this study as well that the more severe patients seem to have a higher antibody response compared with the sort of non-hospitalized, less severe patients. And so again, this is one of those observations that caught a lot of people's eyes as we were moving forward with this, and has been in the back of people's minds as they've started to think about what was going on with antibody responses to this virus. And so that's kind of an idea of the description of what we see with responses to this virus. But there are an awful lot of questions that people have about antibody responses to this virus as well, that we have some data to address, but also that I think that there are many, many open questions to still understand. And the first of those questions has to do with cross-reactive and cross-protective immune responses. I mentioned to you before that we have all sorts of human endemic coronaviruses. There are 4 that are human common cold coronaviruses. They cause about 30% of all common colds. And so you might imagine that there could be a situation where we see different immune responses in individuals who have been previously infected with those common cold coronaviruses and then infected with COVID-19 or SARS-CoV2, as opposed to individuals who might be coronavirus naive and then infected with SARS-CoV2. And when I was making the title for this slide, I sort of snickered to myself because I think that if one thing has been my mantra lately on some of our podcast episodes, it has been that cross-reactive and cross-protective are not the same thing. So just because an antibody is present and reacts, or just because a T cell is present and reacts, does not actually mean that it will lead to protection. And so that's one thing that we need to be very clear on when we are looking at different studies. Is our assay telling us that a particular antibody is a cross-reactive antibody, or is that that assay telling us whether or not that is a cross-protective antibody? And sort of my take home of what I am saying right now, of my discussion of this question, is I think that we have some information about cross-reactive antibodies in the data that has been collected thus far. I'm not sure that we are yet ready to say anything about whether those antibodies are cross-protective. And I think that that is an open question, and I think there are some reasons why that might be really challenging to answer. There have been lots of studies looking at the seropositivity to some of those human endemic coronaviruses. This is one of the more recent studies, but there have been tons. And in this meta-analysis, the authors looked at a variety of different studies looking at seroprevalence of these different human endemic coronaviruses in different age groups, in different countries, using different assays, done at different times. And you can see that the major take-home message is that about 90% of adults are seropositive to most of these, at least in many of our newer studies. There is some variation from virus to virus, but we can assume that the vast majority of adults are seropositive for these viruses. It seems as though everyone is infected in childhood with these viruses. Like I said, they cause about 30% of common colds. And I think that this is one of the places where people have wanted to ask a lot of questions about cross-reactive and or cross-protective immune responses. If we all have so many responses to other endemic coronaviruses, might we expect any cross-reactivity? And could that be influencing what our outcome is with disease? I think one of the reasons why it is a bit of a challenge to address some of those questions about cross-protection is that 90% of adults are seropositive. And so thinking about ways that we can do these studies without adding in the additional variable, say, of age, can be challenging. That we aren't going to find too many people who have no cross-reactive human endemic coronavirus antibodies. But of course, this is something that people are spending a lot of time looking at. This tree shows you the alpha coronaviruses, the beta coronaviruses in their groups, as well as some of the others, gamma and delta. One thing that you can notice is that the beta coronavirus B in orange here is the group that contains SARS-CoV2 as well as SARS-CoV1. And 2 of our endemic human coronaviruses are also beta coronaviruses. They're in sort of this turquoise. You might imagine them being slightly more closely related to our other beta coronaviruses. You can also see that the other endemic human coronaviruses, 229E and NL63, are in the alpha coronavirus group. You might expect them to be slightly less closely related. And there are all sorts of bat coronaviruses that are quite closely related to some of these beta coronaviruses. Here you can see another VirScan study where our COVID-19 patients, again, we've got 232 very skinny columns here, and our pre-COVID controls were examined for antibodies that could react with the severe human coronaviruses, the bat coronaviruses, as well as the common human coronaviruses. And you can see that there is some degree of cross-reactivity. So our pre-COVID-19 controls do have some level of cross-reactivity. They have the most reactivity with the common human coronaviruses, which we presume they may have been infected with. We see that our SARS-CoV-2 patients have considerably more reactivity, perhaps because of the recency of this acute infection. But you can see that there are hits sort of across these groups, indicating that, yes, there are cross-reactive antibodies. And in fact, you could see that even from the epitope map that I showed at the beginning, where you could see that the pre-COVID-19 controls had responses to ORF1 that didn't seem to differ from those of the COVID-19 patients, which sort of indicated that those were some potentially cross-reactive responses. And so, again, we can say that these individuals do seem to have responses that react across some of these different coronaviruses. We do still have some questions about whether or not these responses are going to be cross-protective. In the past couple of weeks, a major study on this front has been published. And full disclosure, the data on this study are complex, to put it mildly. But in this study, there was an examination of antibody responses to different coronaviruses, either the seasonal coronaviruses or SARS-CoV-2 in different patients. And what you can notice is that in the graph that's shown on the left, there are a few different groups of patients, and they are being examined with this novel assay that was described by these investigators for their antibodies that could react with SARS-CoV-2. And they were looking at either IgG, which are shown in red dots, or either IgG, IgM, or IgA, which is shown in a gray dot. And each of these columns, or each of these sort of dots, but think of it as columns, would be a different patient. What you can notice is that in these 2 groups that are shown on the far left, we've got 2 groups of patients who are SARS-CoV-2 negative, and their human coronavirus reactivity was also known. And you can see that some of them do make an IgG response to SARS-CoV-2, again, indicating that there is this sort of cross-reactive response. There also are these patients who are the SARS-CoV-2 positive patients, and they seem to be making this IgG response as well as making other isotypes. And when I looked at these data, I was really struck by the differences between the non-IgG isotypes being made in the SARS-CoV-2 patients, making me wonder about sort of whether or not those types of antibody isotypes may be particularly important in, say, protection or in some of these immune responses. And there is a heat map showing whether or not there is a cross-reactive antibody response to SARS-CoV2 and to all of these other virus types. And you can see that there are some SARS-CoV2 negative adults who have antibody responses, particularly IgG antibody responses to some of those other human coronaviruses. So again, yeah, we know that there is definitely cross-reactive response, and we need to start figuring out whether any of this is cross-protective response. And further, there has been this investigation of whether or not there are differences in some of the seroprevalence in kids who may be getting coronaviruses, those endemic coronaviruses, quite frequently. And it does look like there is higher seroprevalence in children. Why that seroprevalence declines when you've been reinfected over and over and over again throughout life is a little uncertain. But this is the paper that you may have seen in the New York Times trying to explain all the sorts of differences between kids and adults and COVID-19. There may very well be some explanation here with this antibody level leading to some level of protection in kids. And I think that that's, a question that is still very much open. The other big question that comes up when we think about the human endemic coronavirus antibody responses has to do with longevity and quality of those responses. So we know that those responses exist, but the human endemic coronaviruses are thought to be characterized by waning immunity, where people get reinfected over and over and over again throughout life. In fact, I kind of implied that on the last slide. And so we know that an immune response will peak following infection and then will undergo some level of decline. And there is this idea that there is very particularly rapid decline, rapid waning of these human endemic coronavirus responses. Some people have hypothesized that that is due to the location of the respiratory tract and how well antibodies are elicited in those areas, although I have heard many different potential ideas here. Some of this idea about waning immunity came from challenge studies that were done to look at when people could be reinfected with human endemic coronaviruses. A really nice study on this was recently published where a group of HIV patients were studied over time, and their reinfections based on changes in antibody titer were shown. On the left, you can actually see the data from one patient. And so you can see that as this patient had sampling taken at different times, their antibody responses to each of these human endemic coronaviruses sort of came up and went down, implying that they were being reinfected and this immune response was being reprimed. And so you can see, with NL63, there is perhaps an infection here in 1993, maybe another one in 2005, another one in 2007, and so on. And then you can see similar sets of infections with some of the others. This is not characteristic of all viruses. You can look at their measles titer and see that it stayed the same. And when these authors went ahead and tried to quantitate this, they could see the frequencies between different reinfections, and they could see that there were lots of reinfections starting to happen within 6 months in many of their patients there was a particularly large number of them at about 12 months as well. So it does seem like we get reinfected with these human coronaviruses. In order to completely study what's going on with the immune response to these viruses, we have to think a little bit about immune response kinetics. And one thing that we know is that antibody response will peak somewhat after primary infection and will decline over time. The problem is that we've only been studying this virus for a few months. We've only been looking at people's responses for a few months. And so it's sort of hard to tell exactly what this curve will look like and how long those antibodies will last over time. Last week, we talked about these two papers on This Week in Virology. And I will admit that I was a little bit, wasn't harsh, but I made some interesting comments because if you look at the titles of these two papers, they were both just published. One says that the antibodies persist for months, and one says that the antibodies decline over 3 months. And the data from those two studies are shown below, indicating on the left this antibody that persists, and on the right, these antibodies that decline. To me, those both look like sort of the downslope of what we expect of a traditional antibody response. And so I'm not sure that I will fully say that the jury is out or the jury has come back on either of these things. I think that it's still too soon to fully understand exactly what the longevity of these antibodies will be. And I think these antibodies seem to be decreasing at a rate that we might expect them to decrease. This information sort of relates to this question of reinfection and whether or not reinfection is possible with these viruses. Right now, there are 4 cases where reinfection has been confirmed in individuals. And the bar to confirming these cases is incredibly high because we need to have sequence from both infections to show that the virus is actually a different virus causing a second infection and is not virus that has persisted in a long haul COVID type of situation. So there are 4 individuals who have met this bar to indicate that reinfection has happened. 4 out of the number of millions of people who have been infected right now is on the small side. And I think that in some of these cases, you can see that the first infections were generally mild. It's unclear whether all of these individuals actually seroconverted after that first infection, and whether there may have been some other things going on with some of these patients. If the antibody responses were incredibly short-lived, we would be seeing a lot more reinfection we would expect in some areas like in China, where it's been more than 6 months since some of these initial infections. And so I think that is leading people to think that these antibodies may have a bit more longevity than the antibodies to the endemic human coronaviruses, but it's certainly unclear. There's one other kind of thing that is related to this antibody longevity issue that makes me wonder a little bit about what's going on. And that comes from the data that was shown here. This is a really lovely study that actually showed that in the lymph nodes of SARS-CoV2 infected patients, there is a defect in the generation of germinal centers. And so you can see actually quite clearly that in the SARS-CoV2 patients, there are not really any organized germinal centers in the thoracic lymph node, while there are sort of our nice classic germinal centers in our non-SARS-CoV2 patients. And there are big differences in B cell numbers as well as specific B cell subtypes in the germinal centers of these patients, echoing that early observation of reduced B cell numbers. And so these types of data do make me wonder whether we're going to see an antibody response that is sort of typical with this virus. Some of those other proteins like ORF3b or ORF6 or some of the others that we don't really think about when thinking about some of these antibody responses have been shown to be immunomodulatory and to reduce innate immune responses. And so that may influence some aspects of these antibody responses. There have been some other papers coming out more recently that have also said that the B cell response in COVID-19 patients may be a bit unique. Here, there is an indication that there may be some extrafollicular B cell responses that may be reminiscent of B cell responses seen in patients with lupus. And in fact, there have also been a couple of papers that have come out that may say that there is this additional production of autoantibodies in some of these patients. And so questions of whether this antibody response is always positive is really an open question. David asked the question in the chat about antibody-dependent enhancement. And that is another thing that a lot of people have been wondering about. So there is, in theory, antibodies are supposed to be neutralizing and help block viruses from infecting cells. But there have been observations in the past with different viral models that have indicated that antibody-dependent enhancement can be possible, either allowing entry and infection of new cell types, which allows for a different disease state, or resulting in changes in inflammatory cytokines after the virus is able to enter certain cells. There is no evidence of this in any kind of in vivo model with SARS-CoV2. There is some evidence, though very limited, of antibodies allowing entrance into other cells and having increases in inflammatory responses in SARS-1, although those studies are quite limited because of the number of patients that are involved in those situations. There is a well-established description of antibody-dependent enhancement with feline infectious peritonitis virus, FIP, another coronavirus, though one that's not super closely related to the ones we're talking about here. These types of phenomena are right now one of the things that people are looking at really closely in a lot of those antibody studies that I mentioned, but there has been no evidence that has said that antibody-dependent enhancement is happening. That is one of the things that is being watched incredibly closely in a lot of vaccine trials. And when the Pfizer press release came out last week, their press release said this had 90% efficacy in individuals who had never previously been infected with SARS-CoV2. And so it did sort of catch my eye in terms of what happens to people who were previously infected. And so that's one of many reasons why I'd love to see the data. I think that this is that previous antibody presence and whether or not there's any kind of enhancement is something that people are really looking at. But in a human system, antibody-dependent enhancement has really only been shown well sort of in vivo in humans with the dengue model, which is a very different type of virus. So the other thing I will just briefly mention is that, of course, we are now using lots of antibodies in thinking about SARS-CoV-2 infection. And so many of the things we are using for treatment are, of course, drugs that are antibodies either monoclonals to neutralize the virus, like those monoclonals that are coming from Regeneron, convalescent plasma, anti-IL-6 or other antibodies that are influencing cytokine responses, as well as non-antibody drugs like remdesivir. The trick with these, and the reason why I think there have been very disparate results in many of the trials, is that it's not clear that the phases of COVID infection are always carefully examined in treating patients. Of course, when patients are extremely sick, we want to give them a lot of treatments, but that may be past the viral stage when an antibody to a virus may not be as useful. And so that's something that I think that people will, of course, want to think about pretty dramatically. The other thing that I will just quickly mention as well is that there are a number of different vaccine types that are under production right now. Most of them involve just using spike or just using the RBD portion of spike in some type of vaccine modality. The data that have been published thus far from Pfizer, and these are Pfizer vaccine data, Moderna's vaccine data looks quite similar, indicate that patients are making great antibodies in general and neutralizing antibodies to the virus, and they compare to convalescent serum. And so you can see that this vaccine is inducing responses that are sort of on the order of, if not above, natural infection. So that's pretty exciting. One thing that is also potentially interesting is these vaccines, as I mentioned, are largely involving spike and not containing some of the other viral proteins. And so it's possible that we won't see some of the immune modulation and lack of germinal center effect because those other immune modulatory proteins are not present. I hope that was in some way useful for you all in terms of understanding some aspects of antibody responses. I am happy to answer any questions. Thank you all for your attention. I see there's a question from Francis who asks how important I think protection from antibodies is relative to the rest of the immune response. I will first give you a caution, Francis, which is that my PhD was all on T cells, specifically CD8 T cell responses to viruses. And I think that there is some really interesting data on T cell cross-reactivity and potentially some T cell mediated effects. There haven't been as many measurements of T cells in a lot of studies. So I think I am intrigued by some of the T cell responses. I meant to click that. Thanks, Judith. So David also asks, with most of the vaccines in development, which one would I get? I'm not sure that they're all going to end up being available at my local Walgreens. I think that most likely, you know, if you look at the numbers, so there are 9 vaccine candidates in Warp Speed right now. They each are saying that next year they could make 100 million doses. So that's 50 million shots because each one is a 2-dose regimen. The only way we get to enough doses for everyone in the United States is if we have about all 9 of those or, you know, 7 of those. And so I kind of wonder whether it's going to be a geographic thing of California gets this one and Texas gets this one and Northeast gets this one, just to get all of all of the distribution to work out. So I'm kind of thinking I'll get which one is available to me. I'm pretty intrigued by the mRNA vaccines, but once they give me one, I'm happy to have it. So Vanessa asks what I think causes the dysfunctional germinal centers. Vanessa, I would really love to know the answer to that. I lean towards some of the aberrant innate immune responses and aberrant cytokine responses that this virus is driving. The virus is really messing up innate immune cytokines and chemokines during the early days after infection. And so I think that that may prime the B cells and T cells to make improper germinal centers. But I have no data on that. That's my suspicion.
All right, any other questions? So actually I have one more question about it seems like a lot of interest or a lot of work has been done to the antibody response, and we're obviously interested in that, but what about the T cell response? Is it the ELISpot assays too hard to do for people to really look at T cells, or are there other assays that people could do to pay more attention to T cells?
I think part of it is that it's harder to do a rapid T cell examination, but also with T cells, you have the MHC issue where each of us are going to make a unique T cell response. And so in really nailing down a T cell response, you have to pick an MHC type. And so you can say, this is what the T cell response is like in HLA-A2 positive individuals, but it's not going to give you as broad of an answer as some of the antibody studies will.
Right. All right. Well, any other questions? Well, I think I can chime in and say thank you. That was an amazing presentation, and we really appreciate your time. And thanks for chatting with us today.
No problem. I'm happy to answer questions.
All right, good. Well, again, thank you. And we look forward to next month's talk. This will be Aaron Shearer from Emory University. So look out for upcoming emails, and thanks again.
Thank you, David. Thank you, Brianne. Thanks, everyone. Have a great day.
All right.
Thanks.
Bye.